Hello, my name is Dierich Stephan. I'm the CEO of Neubase Therapeutics. We are developing investigational therapies to address Huntington's disease by drugging the genome to address base causality. I'm presenting on behalf of a research and clinical development team that you see here on the slide. Neubase has developed a differentiated platform for both the discovery and the development of novel genome-targeted drugs. These compounds can increase protein expression, decrease toxic protein expression, such as in the case of Huntington's disease, and even edit protein expression in vivo to address root causality, and do so with single base precision, which is fairly unique in the industry. Of course, every disease is genetic, and therefore we hope as a company to have broad impact starting in neuromuscular diseases and then scaling forward from there. We'll be filing our first IND at the end of next year and hope to be filing an IND in Huntington's disease in 2023 using a subcutaneous route of administration that penetrates the blood-brain barrier and knocks down mutant Huntington selectively in the brain, but also addresses the whole body manifestations of the disease. This is a schematic of our platform and how our genetic medicines work. Our platform is named the Patrol platform, and it has two moving pieces. The first is a synthetic DNA oligonucleotide that forms the drug or the pharmacophore. And the second is a proprietary delivery shuttle that we've invented in-house that allows these oligos to be trafficked throughout the body, including into the CNS. Of note, the delivery shuttle allows these neutral water-soluble oligos to traffic directly across the plasma membrane of any cell type in the body and be delivered right into the cytoplasm. Once in the cytoplasm, the delivery shuttle can be jettisoned to leave the, again, neutral water-soluble high-affinity oligo in the cytoplasm, and we can dial in the backbone to have tropism to either transcripts for a particular misbehaving gene of interest, or to the gene itself within the genome. And we've shown, and we'll show data today, that these oligos can diffuse easily into the nucleus and engage the misbehaving gene directly in the duplex genome. The engagement mode can be one of two different binding modes. First is invasion into the duplex genome, which is what we'll describe data on today, and targeting the mutant allele selectively. It's the high binding affinity of these oligos, far in excess of morpholinos and traditional phosphathyroid backbones, that allows this invasion, followed by Watson-Crick sequence engagement. The second binding mode is called triplex binding, whereby the oligo can very rapidly scan the outside of the duplex genome in the major groove and find the sequence of interest and engage. And one can combine these two binding modes to speed up the kinetics while maintaining high selectivity. And the end result is we can modulate gene expression or even engage mismatch repair enzymes to edit mutations out. We won't be talking about turning genes on or editing them today. Now, once a locus is engaged, the activity is conferred through steric interactions with machinery that runs on the rails of the misbehaving gene. And so, for example, when we're targeting the duplex genome, we can put an oligo on the mutant allele and inhibit the ability of RNA polymerase to transcribe that allele and thus form a toxic protein in the first place. If we're targeting the transcriptome, as in the case in the upper right-hand portion of this slide, we can put an oligo on an mRNA and inhibit the ability of a ribosome to either engage or elongate the mutant protein and thus eliminate the toxic protein. So these are the two different mechanisms of action that we'll talk to you about today. As I mentioned, Huntington's disease is the second program that we're maturing as a company, and we're quite excited about and have conviction about the strategy that we're using to get these oligos into the CNS. You're all familiar with the devastating impact of Huntington's disease on mutation-positive individuals, and ultimately our goal is to develop a therapy that is not only effective but extremely well tolerated. And it's one of the attributes of our platform chemistry that, for example, these compounds, peptide nucleic acids, have been shown to be immunologically inert, which of course promises a future where we could identify mutation-positive pre-manifest patients and put them on therapy for years in advance of symptom onset in the hope of pushing out or slowing the progression of neurodegeneration in the CNS and, of course, degeneration elsewhere in the body. And so ultimately that's where we're going with this therapy. We're cognizant that the disease manifests in the striatum, but eventually does spread throughout the brain and cause broad-based neurodegeneration. And thus we want a whole brain solution. We don't want to just bathe the cortex, for example, which is the classic distribution picture after an intrathecal route, nor do we simply want to get drug into the deep brain structures, which is the classical route through intracranial injection, for example. Rather, we want to evenly diffuse drug throughout the CNS by coming across the blood-brain barrier. And of course, every neuron is proximal to a vessel, and thus we hope to have a whole brain solution. But as is commonly forgotten about Huntington's disease, there are manifestations of the disease elsewhere in the body, as illustrated on the left of this slide. And we don't want to forget about those manifestations. And thus we like the systemic route for that reason as well. Of course, all of us have really been thrown by the promising therapies that were recently shelved with respect to intrathecal administration of ASOs, and that is what has and our stance on that is that mechanistically the strategy still holds water in addressing the key pathogenic insult in patients. It's simply that the delivery route was not workable for a large molecular weight genetic medicine, and thus we're wedded to inhibiting production of the mutant protein. We believe concurrently we're also eliminating RAN translation from the mutant expansion, and we are also selectively knocking down the production of the mutant protein while maintaining wild-type protein production. This is a simple schematic to illustrate why from day one we've been so intent on systemic dosing. This is on the left-hand side of the slide, the picture one can expect in terms of biodistribution from an intrathecal route with a large molecular weight compound, essentially reaching high concentrations on the surface of the brain, but not being able to diffuse the 5 or so centimeters into the deep brain structure, which is where the brain first exhibits neurodegeneration. Likewise, intrastriatal injection can get into the deep brain, but not elsewhere in the brain. Whereas on the right-hand side of the slide, you see a representation of why we believe the systemic route is preferable to the other two routes. And of course, while exciting preclinical data has been generated via ICV or intrathecal routes in small-brained animals, you can here hopefully get a sense for the fact that the human brain is 4,000 times the volume of a mouse brain, and it's more difficult to get even concentrations of the drug in the human brain. Now, this is a slide that's meant to illustrate that we really want to very simply knock down as much mutant Huntington as possible while maintaining as much wild-type Huntington protein as possible. And we as a field don't really understand the sweet spot in terms of maintaining wild-type Huntington in the adult human brain. We have a hard time imagining it's not important in some cellular context, whether it's mitochondrial function or otherwise. And to be on the cautious side, our hope is to simply maximize the amount of wild-type Huntington while maximizing the amount of knockdown of mutant Huntington. Ideally, our target product profile is getting rid of 80% of mutant Huntington and maintaining 80% of wild-type protein. So here you see an example of one of our oligos, a conceptual example, and then modules that we can append to it to allow various functions relative to distribution. So tropism for neurons or crossing the blood-brain barrier, extension of exposure to the oligo, ability to get across the membrane, and so forth. And I won't belabor this, but what I do want to exemplify here is that using that delivery shuttle alone, when we inject a therapeutic dose into the tail vein of non-human primates, very quickly, within 4 hours, we see elevated concentrations of the delivery shuttle in the gray matter of the brain, of course, which is more highly vascularized. And that concentration actually increases in all brain regions across the course of 7 days. Drug concentration is illustrated by gray staining here relative to background. Now, the increase in concentrations, or Cmax, over time is particularly interesting in the context of the mechanism. So these drugs are at what are called actively translocated across the plasma membrane of the endothelial lining of the cerebral vasculature and dumped right into the cytoplasm of those cells, and then are then opportunistically exocytosed or transcytosed and dumped into the parenchymal space, where they can then enter neurons once again. And that's a very slow process, but the fact that the blood remains in the circulation and is not biodegraded, and it has a continual amount that's presented to the cerebral vasculature, allows this very slow pumping of drug across the BBB and into the brain. Now, when we connect that delivery shuttle to various payloads, in this case, an oligo that engages the mutant Huntington gene at the DNA level and inhibits transcription, and we test that construct in vitro and in vivo, what you see here in patient-derived cell lines is that with increasing concentrations of compound from 0 micromolar up to 10 micromolar, you see nice selectivity for knockdown of mutant Huntington protein relative to wild-type Huntington protein. And what we like about this is that this persists across several log orders of concentration while not being cytotoxic. And then when we move into a transgenic animal, by subcutaneous injection of the same construct and then harvesting the whole brain a week later, and looking for whole brain knockdown of mutant Huntington versus wild-type Huntington protein, is illustrated on the very right-hand side of the slide. What we see here is statistically significant knockdown of mutant Huntington at the whole brain level. Now, when we move to the RNA level and inhibit translation, as you see on the left-hand side of this slide, by blocking the ribosome from elongating a toxic disease-causing protein, and we couple that to the same delivery shuttle and introduce that into patient-derived fibroblasts in vitro, we similarly see a an allele-selective inhibition of mutant Huntington protein formation relative to wild-type Huntington protein. And when we go sub-Q in the ZQ175 transgenic, we also see at the whole brain level a week later that we knock down mutant Huntington protein selectively and in a statistically significant manner. We're quite excited about this because, as you can imagine, given the biodistribution I showed you, we believe that this could be a very durable effect, especially at the DNA level in the post-mitotic neuron. Next steps, in order to get into clinical trials, we are continuing to work on optimization of potency and selectivity and understand the mechanisms of penetration of the blood-brain barrier by our shuttle, as well as we are currently in life looking for functional rescue in both the R62 as well as the ZQ175 transgenic models prior to moving into PK/PD experiments in higher order mammals, large-brained animals, and exploratory toxicology with the intent of filing an IND in this indication in 2022. So I'll stop there. Thank you so much for having us, and happy to answer any questions.
Hello, my name is Russ Addis. I'm Vice President of Research at NeuExcell Therapeutics. I'd like to start by thanking the organizers at HSG for inviting us to speak to you today. I'm excited to share some of the ongoing research that we have. The topic of our talk is direct conversion of astrocytes to neurons, which we think may be a novel treatment strategy for Huntington's disease. So by way of background, I don't think I need to give much introduction to HD itself to this audience, but as I'm sure you all know, this is a fatal genetic rare disease caused by the mutation in the Huntington gene. But I do want to emphasize that the pathology is really marked by the loss of GABAergic neurons in the striatum that gives rise to much of the motor disabilities that are observed in HD patients. And so although there are quite a few genetic medicines being developed that are aimed at trying to reduce the mutant form of Huntington, NeuExcell is really taking quite a different approach. So we are aiming to regenerate new GABAergic neurons using our novel neuro-regenerative gene therapy, and that's what I'll be talking to you about today. So by way of background, this slide has an overview of various sort of cell and gene therapy approaches that have been taken for both HD and other CNS disorders. And so you can see on the left a lot of different in vivo approaches in terms of using iPS cells to generate new neurons, for example, that could be transplanted. And the concept of direct transdifferentiation or reprogramming that is used to generate iPS cells is really the same sort of concept that NeuExcell is using as we try to directly convert glial cells into neurons. So really what we're talking about is not a cell therapy approach, but really more of a gene therapy approach for regenerative medicine, in which we found that by expressing, in some cases, even a single transcription factor, NeuroD1, we are able to directly convert glia, namely astrocytes, into functional neurons in vivo. Again, by way of overview, our approach is to regenerate new neurons exactly where they are needed within the brain. So at the moment, we're using AAVs. We're using AAV-delivered transcription factors to convert reactive astrocytes into functional neurons in vivo. We have a variety of programs that we're working on, but for the HD program, we're using two transcription factors. What we found is that if we use AAVs to express the transcription factors NeuroD1 and DLX2, that we can generate GABAergic neurons directly within the striatum, right where they're needed. And this is work that has been published by our founder, Gang Chen, and his lab in many journal articles over the years. And what we've seen is that we get a very high conversion rate of astrocytes to neurons, often up to 70 or 80%. We do see integration into existing neural networks, as we'll be talking about today. We see long-range axonal projection to the right target areas. And importantly, we're not in any way depleting existing stores of astrocytes. The remaining astrocytes are capable of cell division and thus can repopulate themselves. So to dive right into data, much of the work I'm going to show you today has recently been published. You can see the reference in Nature Communications in the bottom right of the slide there. So I'm not going to be going into every detail panel by panel, but I do want to at least pull out some highlights from each of these slides. So first, what you're looking at here on the upper left is immunohistochemistry, looking at the striatum of wild-type mice first. And these wild-type mice received injections of AAVs using what we call the GFAP CRE-FLEX system. I won't get into all of the detail of that today, but the bottom line is that by taking advantage of a astrocyte-specific promoter, GFAP, driving CRE recombinase, we can selectively express our transcription factors in the cells of interest, which namely are astrocytes. And what you can see is that in addition to the transcription factors, you can see a little schematic in the bottom right there, panel D, we're also expressing a reporter gene, mCherry, so that we can monitor the transduced cells over the course of the reprogramming process. And if you look early in the reprogramming process, 7 days following injection, as you can see in panel A on the upper left, the vast majority, if not all, of the reporter-labeled cells are also GFAP-labeled, meaning they are astrocytes, and this is what we would expect. We know that our vectors are driving transgene expression in astrocytes. But if you look later, in this case, 30 days after injection, now you see quite a different result. You see a loss of the GFAP astrocyte marker, and now you're starting to see that these label cells express NeuN, the neuronal nuclei marker, a marker of mature neurons. And so what we're able to see, and you can see in that panel C there, is that in the presence of both NeuroD1 and DLX2, although at that early time point you're seeing mostly GFAP positive cells, by that 30 days after injection, you're primarily seeing neurons. This, we think, is indicative of reprogramming of astrocytes to neurons, and we'll talk more about that. So this slide is showing more of a time course, and so looking at this process over time, you can actually visualize by looking at these different time points, the steps along the way. And so in the panel on the upper left, you can see in control-treated mice, so these mice received only AAVs that express the mCherry reporter gene, that GFAP expression over time in the labeled cells remains roughly unchanged. You don't really see much expression of any neuronal markers, whereas if you look in the bottom left panel with the expression of NeuroD and DLX2, you see a dramatically different result. You see in blue, the blue line, progressive loss of that GFAP marker, indicating that cells are losing their astrocyte identity. And on the green line, you see this steady increase of the neuronal marker, indicating that these cells are, in fact, fully being reprogrammed into neurons. And in the panel on the right, you can see that they're becoming the right type of neurons. So we want to generate GABAergic neurons. We can see those the first two markers there, GAD67 and GABA, are indicative of GABAergic neurons being formed. And then we also want to know that we're forming the right subtype of GABAergic neurons, in this case, the medium spiny neurons that are heavily implicated in HD pathology. Those are recognized by the marker of DARP32, and we see that the great majority of neurons that we're generating are, in fact, consistent with medium spiny neurons. The other markers that you see present are would be found on other types of interneurons, and so as you can see, we are not generating those types of neurons. We really are specifically generating medium spiny neurons in the striatum. So now looking at a little bit of histology here and shifting gears, the previous data that I showed was in wild-type mice. Now, of course, we want to move into some Huntington's mouse models. And so first I'm going to show the next few slides are using the R62 mouse, a very severe, a model of very severe HD, very, very rapidly progressive. And even grossly in terms of the histology, you can see that this enlargement of the lateral ventricles due to tissue atrophy in the striatum that you see in the control-treated mice, that is somewhat rescued in the mice on the right that received NeuroD1 and DLX2. And then again, on the right side with some more quantitative data, as I showed previously, we are generating the right type of neurons. We see markers of GABAergic neurons and of the MSN subtype expressing DARP32. moving on, we're starting to see some of the more exciting results that came out of this project. We're really starting to see rescue of the pathological phenotype in that R62 mouse brain. As you look through, if you're moving through various coronal sections here, you can see striatal atrophy in the control-treated mice up top, whereas you see a reduction in this atrophy in the mice that received the NeuroD1 and DLX2 AAV vectors. And this is quantified on the right, where you do see statistically significant rescue of this striatal atrophy in the R62 mice. Of course, always very interested in functional recovery, and we're happy to report that we see that as well. So what you're looking at here are a couple of different assays. It's always challenging to model a human disease in mice. I think we all know that. But fortunately, the R62 mice have been very well characterized over the years for some of their different functional endpoints. So I'm showing two of them here. First, looking at the stride length. You're looking at each little dot in the upper panel is looking at a different paw touching a spot on a grid. And what you can see quantified in panel B on the right is that you do see the R62 mice in that gray bar have a reduced stride length compared to the wild-type mice in black. And control-treated R62 mice are no different from the parent R62 line, whereas with NeuroD treatment, we do actually see improvement, statistically significant improvement in stride length. And on the bottom panel, a different assay looking at really just the amount of distance that these mice walk. You can see the wild-type panel in the upper left, this tracing the activity of mice throughout a cage on a particular grid. That you can see quite a bit of activity, whereas the R62 mice and the control-treated mice have dramatically reduced distance that they travel. Whereas in the bottom right panel, you can see that with the treatment of NeuroD1 and DLX2, you do see this functional improvement, and that's quantified with the blue bar on the right graph in panel E. Also wanted to look at lifespan. The R62 mice do have a significantly reduced lifespan versus wild type, and here you can see that over the course of a 100-day long assay, we see improved survival, improved lifespan in these mice. You can see that it was really, the treated mice, almost the great majority of them did survive this 100-day assay, whereas nearly half of the untreated mice or control-treated mice did not. So very, very excited about that result. Of course, very interested in looking at aggregates of the mutant Huntington protein, and so we've looked at that by immunohistochemistry here. And what you can see in these panels, the green dots are looking at Huntington aggregates. And we thought it was very interesting. We really didn't know necessarily what to expect. As we're starting with astrocytes that presumably all carry the same Huntington mutation. So we wanted to see, would the neurons that result from reprogramming these astrocytes also express Huntington? And we do see reduced levels of these Huntington aggregates in the newly formed neurons. Exact mechanisms of why this might be are something that we're still very interested in figuring out, but it's certainly encouraging and gives us some confidence that this might really be a viable therapeutic option moving forward. Last bit of data, just wanted to show in a different mouse model of Huntington's. The previous work was using the R62. This is now using the YAC128 mice. These are a more slowly progressive model of Huntington's disease. So you have to really look in aged mice, and this is looking in 15-month-old YAC128 mice, simply to show that the reprogramming process works here as well, in addition to the other mouse models that we've shown. So really just giving us greater confidence that this approach can generate new medium spiny neurons, GABAergic neurons in the striatum in multiple mouse models now. So to summarize, so far, we've shown really efficient in vivo conversion of striatal astrocytes into GABAergic neurons. This is expressing two transcription factors, NeuroD1 and DLX2. And it seems that this process takes 3 to 6 weeks or so, so fairly rapid. In terms of the markers that we see, we're seeing really all of the right markers for MSNs within the striatum. Certainly very excited about that. And perhaps most importantly, we're really seeing an improvement in both neurodegeneration, some rescue of phenotypic defects, and extended lifespan in a mouse model of severe HD. I think that we've also been able to show conversion in a slowly progressing HD model, as I showed on the last slide. So a relatively recent development for us that we're really very excited about is in August, we signed a research collaboration agreement with Spark Therapeutics. As many of you may know, they're really a world leader in gene therapy. And Spark is going to be working with NeuExcell to accelerate the development of this gene therapy approach to treat Huntington's disease. So we've really just kicked this off. Both sides are really excited and eager to work together, and so we'll look forward to reporting back to the HSD as we make progress on this. So I'll close there and say thank you. Once again, NeuExcell is really excited to be leading what we think is a gene therapy effort for neuroregeneration and repair by converting astrocytes into neurons. So look forward to having a conversation and discuss this with you. Thank you.
Hi, I'm Doug Mcneil, Vice President of Preclinical Development at Ophidian. We're developing technologies to treat neurodegenerative diseases, in particular, platform technology to deliver ASO siRNA into the brain after IV delivery. And I'll talk a little bit about the technology, some of the data we have, and our current progress preclinically in developing a therapy for treating Huntington's disease. So the treatment of neurodegenerative diseases, as you know, is complex. We have the blood-brain barrier to address, which keeps most molecules out of the brain, and certainly almost all large molecules and all highly charged molecules. And it's also particularly multigenic, leading to difficulty to select a single therapeutic pathway. Current practice is to deliver therapies, especially large molecules, intrathecally or even intracranially. And then the brain is reached through diffusion out of the CSF from the ventricles. And this can limit therapeutic therapies reaching deep brain tissues. Now, what we've developed is what we call a Trojan Horse. It's a peptide that can be delivered IV, allowing systemic delivery, and it reaches the brain through the blood-brain capillary network. And so it transports across these capillaries into the brain. Thus, we should be able to reach any area of the brain and allowing widespread uptake of molecules into the brain. We're approaching treatments by looking for validated monogenic disease targets. And of course, Huntington's disease is an obvious candidate. And we're trying to silence expression of those genes using siRNA or ASOs that are complex to this Trojan horse peptide. And essentially, we use the peptide as to hitchhike molecules into the brain. Now, this is a cartoon that kind of shows how this works. OCCT is a peptide. Depending on the variant we have, it can be 20 to 30 amino acids long. It's a synthetic peptide designed based on an allosteric site on the nicotinic acetylcholine receptors. And we've looked at a number of proteins, peptides, neurotoxins, viruses that bind to this site and selected a consensus peptide that will allow us to bind at a modest affinity to this allosteric site and hitchhike along with the normal physiological processing that occurs for the receptor. Now, the peptide OCCT has been designed to bind to the two subtypes of the nicotinic acetylcholine receptor that are predominant in the brain, the alpha-7 and alpha-4 beta-2 subtypes, while having lower affinity and not being targeted to the forms of the receptor that exist in the periphery. And so in this way, we're hoping to preferentially deliver molecules to the brain over other tissues in the body. Because it's a peptide, we can deliver molecules two ways, covalently, just by linking them, or, which we'll be talking about today, electrostatic, in which you use addition of polyarginine tail to the peptide with its positive features to bind to the negative charges on either siRNA or ASO. And essentially, we now form a small nanoparticle which has been neutralized the charges of both molecules. And as a neutral molecule, it's easier to move through circulation into the brain. And it's still the OCCT that binds to the receptor. And cartoon on the right kind of gives an example of that. We show the complex listed as D in the triangle binding to the nicotinic acetylcholine receptor on the endothelial cells of the blood-brain barrier. This receptor normally goes internalization and then recycling to the surface of the cells. And it can recycle to, presumably with a 50-50 chance, to the blood side or the brain side. If it recycles to the brain side, our OCCT and drug cargo can be released because of their low affinity binding into the brain, where they then interact with receptors on neurons. In this case, they get internalized again, just trafficking along with the receptor in its normal physiology as it binds the agonist. And once internalized into neurons, a fraction of these receptor complexes will enter the lysosome. The lower pH will lead to dissolution of the electrostatic interaction and release to endosome escape of the siRNA into the neuron, where being highly charged, it's now trapped for long periods of time, can undergo multiple catalytic cycles and have a long-term effect in knocking down message. Now, this means that we can use a single OCCT type molecule to deliver a number of different cargoes. The sequence of the siRNA, sequence of the ASO are not particularly different as long as the charges are approximately the same. And so once we validate the platform OCCT with one target, it should be available to deliver gene silencing for other targets as well. So the supporting data we generated to support that this target platform technology of OCCT is efficacious is we looked at gene knockdown in vitro in cell lines using either primary neurons or neuronal-like cells. And we've demonstrated knockdown of a number of disease targets involved in Huntington's disease, Parkinson's, Alzheimer's, and cognition. From those cell line studies, we select effective complexes and move on to mouse models. Generally, we'll go into wild-type mice first because part of our platform is we select siRNAs that have affinity for both the rodent and human gene target, simplifying drug development. And we've demonstrated knockdown of message for Huntington's disease, which I'll show you a significant amount of that data. And we've also shown reduction of a gene involved in cognition called LINX1, and we're able to knock that down in wild-type mice. And then we move on to efficacy models, typically humanized mouse models of Huntington's disease. We've been able to demonstrate knockdown of mutant Huntington mRNA. And we've also determined that after knockdown of LINX1, we see an improvement in memory, indication improvement in cognition. And similarly, in mouse models, we've demonstrated a reduction in pain with a neuropeptide delivered that's involved in the sensation of pain. So in multiple gene targets, we're able to knock down both in cells, wild-type mice, and at least for Huntington's, in humanized Huntington mouse models. So now we'll be talking about specific data from our Huntington's disease program. So this slide summarizes the studies we've completed, and I'll talk a little bit about the data. As I mentioned, we used human cell line to identify multiple siRNA and ASO candidates. We then went into wild-type mice to select the optimal combination of OCCT and a particular siRNA. And since then, we've been studying the knockdown of mutant Huntington mRNA in the back Ph.D. mice following a single IV administration. And as we'll show the data, we've been able to get about a 50% reduction in mRNA in a variety of tissues, and the knockdown has lasted after a single dose for up to 4 weeks. We're now in the process of a chronic study to look for changes in Huntington disease phenotypes in CQ175 mice. So far, what we've done is after 2 doses, confirm knockdown of Huntington mRNA in various tissues. Now, as part of a platform technology, we've also done some additional studies, such as showing a radiolabeled study with and Irwin behavioral test, and cytokine induction studies, which I'll also summarize quickly at the end. So as I mentioned, after cell line studies, we move into wild-type mice, and in a short screen, it was 3 days post-dosing, we look for changes in Huntington mRNA with multiple complexes, some siRNA, some ASO, leading us to select a particular complex which showed good knockdown in both striatum and hippocampus. This particular complex, which we designate OCCT1, siRNA3, exists at a 10-to-1 molar ratio, which is approximately what you'd predict is needed to neutralize the charges on the arginine residues on the OCCT and the phosphate backbone on the siRNA. And at this molar ratio at a single IV dose, we can see knockdown in the hippocampus and striatum at both day 3 and day 7 after the single dose. So this is good enough data for us to move on and initiate additional studies. So one of the studies, of course, is how frequently do we need to dose our complex IV to maintain knockdown of message in the brain? In this study, we looked at the level of knockdown 1 week, 2 weeks, 4 weeks, and 8 weeks after a single dose. The dose here is 50 micrograms of siRNA, or about 2 mpk of the complex. And we can see we get knockdown after 1 week, 2 weeks, and 4 weeks, and it pretty much is starting to return to baseline by 8 weeks. Recently, we've also been assessing tissues for knockdown of Huntington protein. So in this particular study, again, after a 50 microgram dose, we saw a knockdown at 2 weeks significantly, as well as 4 weeks, not statistically significant. But then we looked at the message or protein levels in those brain samples, and again, we saw a knockdown of between 20 and 30% from 1 week to 4 weeks. So we are seeing knockdown of both message and protein after a single dose. As I mentioned, we've begun a chronic study in the Q175 mice. So we assessed a knockdown in this model as well. And here we report the knockdown in striatum and hippocampus, or striatum and cortex, 2 weeks after having delivered 2 doses. And here we see we're getting about a 40-45% knockdown in both tissues. On the 2 color bars, blue and orange, represent 2 different primer pairs. And saline control, or vehicle control on the left, just to show that it is specific to our complex. And the gray bar is another neuronal gene, again, demonstrating we're knocking down specifically the Huntington genes and not just genes neuronally expressed. So as I mentioned, part of the platform is to hopefully demonstrate its utility and safety. So we looked in an Irwin behavioral study. In this study, mice are injected with the complex, and then over a period of time, between 30 minutes and 24 hours, they are assessed for behaviors that are associated with known drugs that affect the CNS system. So various behaviors in the mouse, everything from hyperactivity to lethargy, to just not eating, a variety of behaviors. And the point was, compared to the vehicle-treated mice, there were no treatment effects observed due to injection of our complex IV. Similarly, we looked at the ability of the complex, or hopefully inability of the complex, to induce cytokines. Certain forms of siRNA can induce cytokines in the liver. And so we looked at dosing vehicle and our complex, and after 2 hours, isolating plasma to look for any evidence of induction of cytokines. And among the 10 pro-inflammatory cytokines we looked at, there was no difference versus vehicle. So kind of giving us some confidence that this peptide platform is likely not to have any serious, obvious, it at least has no obvious safety issues at this early stage. And of course, if our theory is right, that binding to our peptide, which has been selectively targeted for neuronal forms of the nicotinic acetylcholine receptor, we would expect enhanced uptake into the brain and reduced uptake into peripheral tissues. So in this case, we labeled siRNA with tritium, formed a complex, gave a single injection, and then measured the radioactivity in the brain and liver over time. And gray line, it's just the radioactive siRNA alone as a baseline. And compared to that, there's significant increase of uptake in the brain, about 2 to 3-fold, and a significant decrease of uptake into the liver, similar. So it appears that our brain targeting does seem to be effective. So our ongoing studies, I mentioned, we're looking at a chronic study in the Q175 mice that we hope to finish later this year. We're looking for changes in Huntington disease phenotypes, including behavioral phenotypes, levels of HTT protein in CSF and brain, changes in neurofilament light chain, CSF and plasmid, and the formation of aggregate Huntington protein complexes in the brain. And so this will all be compared to vehicle-treated mice, and hopefully we'll see an improvement in these phenotypes. If all goes well, we'll hopefully then begin IND-enabling preclinical studies and potentially submit an IND in fourth quarter of 2022 or first quarter of 2023. And I just want to point out of the Ophidian team, all the mRNA work that I described today is the work done by Brigitte Angenou at our lab in Pasadena. And I also want to point out that our CSO and founder, Julie Miwa, was the one who came to the discovery of this potential allosteric site as a means to target delivery to the brain and help develop the OCCT peptides that we're using in the lab today. Thank you.
Good morning, good afternoon. Thank you very much to the HSG for inviting Triplet to give an update on TTX-3360, our antisense oligonucleotide that we are developing to prevent onset and progression in HD and other repeat expansion disorders by halting somatic expansion. As part of today's presentation, I will be making forward-looking statements. This is the outline of my presentation. I will briefly go over the background of somatic expansion in repeat expansion disorders and the role of DNA damage response genes. I will then present the way how we have selected TTX-3360 for development in HD and other repeat expansion disorders. And I will end with some really recent data from SHIELD HD, our natural history study, and how this data will inform our phase 1/2A clinical trial in HD starting in mid-2022. So repeat expansion disorders are a growing number of diseases that are characterized by a mutant gene that carries an expanded short repeat sequence. So each disease has a unique disease gene, but there is a common mechanistic feature of those indications, and this is what we call a two-step hypothesis. So inheriting the disease gene gives you the predisposition, or gives an individual the predisposition to develop a disease such as Huntington's disease. But then there is a necessary second step that is involved in somatic expansion. So an expansion not in the germlines, but particularly for the CNS repeat expansion disorders in neurons in the brain that occurs in life and is a critical trigger for disease manifestation as well as progression. And thanks to the beautiful data from Jim Gazella and his team at the MGH, they have established genetic modifiers that affect onset as well as progression of repeat expansion diseases, and in particular Huntington's disease, by modulating somatic expansion. As you can see here on this graph, on this Manhattan plot, this is a follow-up Manhattan plot from the initial data that showed a strong impact of DNA damage response genes on age of onset in HD. But this is a further data set that Jim presented at the April CHDI meeting this year, where he has shown that these same genetic modifiers also affect the progression using the total motor score or the symbol digit modalities test, so a cognitive outcome measures. And what you can see in particular is that for the symbol digit modalities test, that MSH3 is clearly linked to deteriorating in cognitive features measured by the SDMT, but also for the TMS, MSH3 is clearly associated with a deterioration. So this also indicates that while many DNA damage response genes have been sort of identified as genetic modifiers, MSH3 is particularly interesting given that it really clearly affects also the onset, but also the progression of the disease. Now, the next two slides just compare and contrast some of the key differences between targeting the disease gene, and so kind of like step one in this disease process, and targeting a genetic modifier such as MSH3, so step two in this disease process. So targeting Huntington, in particular mutant Huntington, will ultimately not abrogate somatic expansion. And if we don't abrogate somatic expansion, there's continuous somatic expansion, which will ultimately lead to the more toxic exon 1 production, but overall to an ever-increasing toxicity of the mutant Huntington gene. This might explain why animal data show that at least 75% mutant HTT lowering is needed to show a therapeutic benefit. Obviously, targeting mutant HTT is only applicable to HD, and looking at our own data as well as published and presented data, we have concluded clearly that an intrathecally delivered unconjugated ASO does not deliver to the deep brain areas such as the striatum, which of course is affected very profoundly in HD and also very early on. Given the critical role of HTT in neurodevelopment, there is also a concern intervening in young patients and potentially also in pre-manifest patients. Now, looking at targeting a genetic modifier, and in our case, in particular MSH3, we know from our data, but also other people's data, including in particular Vanessa Wheeler's data, but also the human genetics, that 50% lowering of MSH3 halts somatic expansion. By halting somatic expansion, we abrogate this ever-increasing toxicity of increasing expansions, and it is assumed that we will also prevent the production of the ever more toxic exon 1 fragment. As I said, 50% is sufficient for this effect, and because it is the step two in the disease process, is this upstream of a disease gene, so it is potentially targeting more than 30 CNS repeat expansion disorders with the same molecule and the same route of administration. We have decided to use the ICV, the intracerebroventricular route of administration, because, as I will show you as part of this presentation, with this route of administration, we can get target engagement, so lowering of MSH3 profoundly in both the striatum and the cortex, and achieve this 50% lowering, particularly in the striatum, where we know that somatic expansion occurs early in the disease process. And because MSH3 has not been involved in any particular developmental aspects, there is no concern targeting pre-manifest patients nor younger patients. Now, as we then think of developing our particular antisense oligonucleotide and how we came to select TTX3360. So first of all, There have been multiple genetic modifiers that came out as significant modulators of onset and, as you have seen before, disease progression. However, when we then look at their potential safety profile, and this is here using the NOMAD database, this is genetics in healthy people, and it tells you whether there is a loss of function tolerability and whether this gene is highly constrained or not. So the more to the right a gene is in this graph, the more tolerant it is to loss of function. And of course, by lowering a gene by 50%, we want to have a good tolerance to partial loss of function. And you can clearly see that MSH3 is much more shifted to the right, so shows much greater tolerance to loss of function than some of the other DNA repair genes, but most importantly also much better tolerance to partial loss of function than the disease gene itself, particularly for HTT, but also ataxin 1 and 3. And we know, of course, that HTT knockout is embryonic lethal, and we have seen that MSH3 knockout mice have no phenotype and a normal lifespan, as you can see here, not different from the wild-type animals, also no difference in the tumor burden compared to the wild-type animals, where again, for some of the other DNA repair genes, there is a shortened lifespan and a higher tumor incidence. And we know, of course, particularly in humans, that MSH2 mutations are associated with Lynch's syndrome. So given the totality of the data, MSH3 really stands out as a gene that shows clearly efficacy in the human genetics and the animal model data, but shows also a profile that indicates a good safety profile when lowered by 50%. So then we went about to designing, so Brian, my colleague, went about to design ASOs that has little off-target effect based on the in silico considerations. We tested them for in vitro potency in multiple different cell lines, then for in vivo tolerability, both in rats and mice, and ultimately in non-human primates for safety, tolerability, PK/PD. Based on the totality of the data, TTX3360 was selected as a clinical candidate. It shows very sustained and very good target knockdown of MSH3 in both the striatum and cortex, and I will show you the data shortly. It has been safe so far based on clinical observations in more than 90 non-human primates, including repeat administration ICV. And we have not seen any NFL increase when we administer TTX3360, either IT or ICV. I mentioned before that we have evaluated the route of administrations and have selected ICV because with an IT injection of an unconjugated ASO, there is clearly very, very little distribution to the brain, while there is a lot of distribution in the cortical areas. But again, the striatal areas are of course key as we think of targeting HD patients. When we give an ICV injection of TTX3360, we see very good exposure in the caudate as well as the putamen. And so this is why we have selected the ICV route of administration. And based on more recent, even safety considerations, we believe that ICV administration of an ASO is much more sustainable long-term because it uses a device that once implanted allows a convenient re-administration of the drug. So the next slide here shows you non-human primate data with TTX3360 injected into one lateral ventricle. And you can see here the mRNA for MSH3 normalized to 5 housekeeping genes for most robust data. And you can see here that over 12 weeks after a single injection, we can measure the knockdown very nicely in the caudate, putamen, and accumbens, as well as in the cortex, both the frontal and the temporal cortex. And what you can see here that in the case of the putamen, while it takes some time for the ASO to actually reach the putamen and then induce the target knockdown, at week 12 post a single injection, we still see about 50% knockdown, and we anticipate that maybe there's even a longer knockdown than we have currently been able to measure, but we are doing studies where we look even at 24 and actually even 48 weeks post a single injection. Now, these data, of course, bode well for as we think of the clinical trial that we plan to start in mid-2022. TTX-3360 is currently in IND-enabling studies. But we have also launched SHIELD-HD, a natural history study that we will use to prepare the design for our phase 1 clinical trial. So SHIELD-HD was started in 2020, and we thank all participants, patients and investigators and all site staff for an exceptional recruitment. So we have recruited 70 patients within about 6 months in 2020. The patients were recruited into 3 cohorts based on a range of CAP scores. So cohort 1 and 2 are more pre-manifest patient population, and then cohort 3 is an early manifest population. Patients show a representative HD patient population with the age range and the mutant HTT-CAG repeat numbers. And we will use the SHIELD-HD data either as informative run-in for those patients who roll into the phase 1 after having been in SHIELD-HD, but we can also use the SHIELD-HD patient data as an augmentation of the control group of our phase 1/2a clinical trial. And we have discussed this approach and got support from different regulatory agencies. Now, this is the data that is the first longitudinal data in SHIELD-HD. So it's only 16 weeks. So, of course, this is short for an HD trial. And we are thankful to Jeff Long for producing these analyses. And so what you can see here again in these 3 cohorts, cohort 1 and 2, pre-manifest patients, cohort 3, early manifest patients. And you can see here that over only 16 weeks, we see directionally a change, a worsening in the components of the composite UHDRS as expected. And so this is, of course, very nice because this data will be critical, particularly once we have 48-week data, which we will have later this year. And so by Q1 2022, we will have data from our SHIELD-HD data over 1 year, and this will allow us to make an informed decision for some of the particular outcomes that we want to assess in the phase 1/2a proof of concept trial. Based on an initial trial simulation using the coded volume, Jeff Long has evaluated that with 80% power for only a 30% treatment effect, we can detect a significant effect with 60 patients on active treatment over 18 months. And so this kind of trial simulation based on the SHIELD-HD data will be very relevant for our phase 1/2a trial design. We have also measured CSF NfL. You can see here again very much in line with the data published by Ed Wild and Lauren Byrne in HD patients, and no change over a 16-week period, which is also in line with prior publications. And interesting here, we have also included the HD cognitive assessment battery in SHIELD-HD, and we can see here very nicely a negative correlation between CSF NfL and the HD CAP at baseline. But SHIELD-HD has a second very important purpose. We are collecting CSF samples at screening or baseline, week 16, and then week 48, and then also at week 96. So this CSF samples are very, very important for the optimization of a target engagement biomarker assay that Peter at Triplet Therapeutics and his team are developing. The team has made wonderful progress. They have developed a solid protocol for the EV isolation, extracellular vesicle isolation from human CSF. And this is actually data from one HD subject in SHIELD-HD. And so this is how a normal AV pattern looks like in the CSF. And we have done qPCR for the mRNA of MSH3, and we can see that we detect moderate levels in the CSF, in the RNA extracted from these EVs. And so we are doing more optimization so that we are ready with our target engagement biomarker assay at the time of our phase 1 clinical trial to be sure that we can measure target engagement. Now, this data set is a proof of principle So we have here transfected human cells with TTX3360. And what you can see here that the knockdown that we can measure in cells, either at 24 or at 96 hours post-transfection, is very nicely recapitulated at the mRNA reduction level in the EVs, in the supernatant of the cell culture system. So again, very nice correlation between tissue knockdown and EV knockdown. And we will, of course, be doing more experiments in non-human primates to corroborate this in vivo. And with this, I come to an end. So Triplet is based on the hypothesis and the data that support this hypothesis that manifestation in Huntington's disease and other repeat expansion disorders is triggered by somatic expansion of the mutant disease gene. Others and ourselves, we have shown that 50% lowering of MSH3 provides a strong effect by halting somatic expansion. We have shown you that the ICV route of administration is a very robust way to deliver an ASO to the key brain areas, such as the striatum in HD, and induce the knockdown that we need, which is 50%. And we believe, based on the data we have seen from other programs, that repeat ICV administration is more sustainable long-term than repeat IT administration. I have shown you the nice biodistribution in non-human primates, which indicates that we could have very convenient clinical dosing due to the long duration of action. So far, we have not seen any NFL increase, but we have seen a good safety in terms of clinical observations with TTX3360, and we are on track for CTA IND submission in mid-2022. And with this, I would like to thank you for your attention, but I would like to thank our many advisors and collaborators, as well as all the patients who have provided us feedback and who are, of course, in SHIELD HD as well. And I would like to thank also a fabulous team at Triplet Therapeutics that has worked very hard, is continuing to work very hard to deliver TTX3360 to patients by mid-2022. Thank you, and I will be happy to take questions.
Good afternoon, good morning, wherever you may be. Thank you for joining us today. My name is Brian Pfister. I'm the Global Clinical Lead for Huntington disease at PTC Therapeutics. We're incredibly excited to be here today to talk to you about what we've been doing with our program and give you just a quick clinical update. As you may know, PTC has been around for the better part of 3 decades. We're starting our 3rd decade here, so about 24 years. We've been in multiple disease states. We've brought drugs to market in Duchenne muscular dystrophy. We've had a lot of success with our splicing platform. We're really dedicated to bringing treatments to market that patients haven't had in the past and really helping to bridge this gap of unmet needs for patients, and HD is no different. So you can see we've got things in gene therapy, our BioE platform, metabolic platforms, oncology and virology. But really what I want to talk to you today about is the splicing platform. So the splicing platform has been spearheaded by the Erisdi program. So risdiplam is the generic name for spinal muscle atrophy. So we've really been able to drive innovation in this space. And from this, we feel confident that we can move into HD by using the splicing platform with regard to the Huntington protein. So we're incredibly excited about this. This is a huge breakthrough for us and a huge innovative move for us as well. So in order to do this in HD, we feel it's really, really important to apply the splicing technology at the root cause of HD. So this means we're going to be targeting Huntington. So this is a Huntington-lowering small molecule. What we understand and what we believe in the time that we've been working on this is that there are certain aspects of the program which are critical to success for the patients and outcomes in HD. First is we have to treat HD as a whole. This means broad tissue distribution. So this means both in the CNS and periphery. We want to make sure the drug is getting where it needs to go. We want to make sure that the drug is orally bioavailable, penetrates the blood-brain barrier, and is in the target organ of engagement. We want to demonstrate the uniform HTT lowering in the brain. So this is important in key areas such as the striatum and cortex. The other component we really want to address is really HD-associated peripheral dysfunction. So this is skeletal muscle, liver, immune systems, just to name a few. And we really think it's important for the drug to be reversible and titratable. PTC 518 has been developed with this end in mind. So when you think about it, PTC 518 is built, designed, and developed specifically for HD, as compared to some other products that may be retreads or uses from other disease states. We are looking at this head on with HD in mind. And so what this means is at the beginning of this road to success, we really have to find a number of molecules which we believe to have the qualities, as I mentioned on the previous slide, have the qualities to be beneficial in HD. So this means looking through the molecules that we have, understanding what the needs are in HD, and then looking at drugs, once we identify them, looking at drugs that we know will have HTT downregulation in cells. So we want to understand, is it really going to engage the target that we're hoping? From that, we can identify the molecules which fit that bill. After we have identified the molecules that fit that criteria, we want to look at this in the preclinical models. So we want to make sure that we do have HTT downregulation. We always start with mice, poor mice, but it's definitely good to inform us of what the process is going to be. From that, once we find the molecules which are capable of lowering HTT, we want to make sure, and this is incredibly important, that the potent analogs are available and they have reduced efflux. So this means once we get into the target tissue, we want to stay in there. Getting in there and being effluxed out or being pushed out is not enough, in our opinion, with regard to Huntington disease. From this, as we've done these other steps, then we were able to find PTC 518 and move it forward into the clinic. And so what this means is we start out in healthy volunteers. We want to understand the pharmacokinetics and dynamics of the product. So we start this in healthy volunteers. From that, we see that the Huntington RNA is reduced in healthy volunteers, which is a critical milestone, because this indicates to us that when mRNA changes and goes down, protein will follow suit. This allows us to move forward into the PK/PD section of the program in HD patients. So we've moved from healthy volunteers now into HD specific patients. From this, we're able to build on success and grow into the clinical efficacy in patients and ultimately, make it available to patients. So we're incredibly excited about the process. It's a long road. It's very detailed. It's very specific. And again, 518 is designed to really address the issues that we see in HD. We've looked at splicing modifiers in the past, and so not specifically 518, but we've looked at other splicing modifiers. And what we've seen is there is a dose-dependent in vivo HTT lowering. So you can see here in the graph on the left-hand side of the screen, you can see that as we increase dose, we have decreased the amount of HTT remaining. So this is important to us because this is, again, making sure the target of HTT can be lowered from an mRNA standpoint and a protein standpoint, but we also want to make sure that we see that the areas of the brain we have uniform lowering. So we would have striatum, cortex, and cerebellum all being lowered effectively with the splicing modifiers. This is important, as I mentioned before, because we want this uniformly lowered across brain regions to maximize the treatment effect. So when we look at 518 specifically, I showed you previously splicing modifiers. When we look at 518 specifically, what we see, and you can see at the time points from 0 to 24 across the bottom, and then percent of Huntington RNA lowered from baseline, what you see is as the drug comes up in concentration, the HTT RNA levels in blood go down. This is incredibly encouraging for us because this confirms that 518, just like the other splicing modifiers, has been able to lower the HTT RNA in blood. What we do is when we overlay this onto the PK profile, as we see the drug coming up in concentration in the blood, then we see the correlation with the decrease in the RNA levels. So the drug is actively in the blood, the drug is actively then lowering HTT RNA levels, which would indicate to us as RNA goes, so too does protein go. We would completely expect to see Huntington lowering occurring with this drug. This is great in the preclinical model. This allows us to move forward into other models to look at and to understand, and ultimately then into healthy volunteers. So when you look at this, as I mentioned, that the protein levels in blood and the protein levels in brain, we want to make sure that the molecule is able to lower both. So just blood but not brain isn't enough. And so what we see here is the 518 molecule in the back HD mouse model. It definitely shows that. So you see the lowering that occurs in the blood, and it have about the same lowering that occurs in the brain. So this is critically important to us, and this is, again, a major hurdle that we've just overcome. So this helps us to understand the molecule will do what it's supposed to do, and we can have confidence in the design as we move forward with the program. So this allows us, as I mentioned, this is the healthy volunteer study, and it's really a phase 1 done in 4 parts. The first part is a single ascending dose, so this would be 1 dose given, and then we look at the pharmacokinetics and dynamics. We did 5 cohorts of 8 healthy volunteers. The endpoints here, we're looking at HTT mRNA splicing. From there, we can understand what we need to do going into the multiple ascending doses. So this would be up to 5 cohorts of 8 healthy volunteers. We evaluated the safety and tolerability of HTT mRNA splicing and looking at protein. So again, it's important to see both of those components because they go hand in glove. Then we looked at food effect. We want to understand if there's a food effect with regard to the drug, parallel design, and looking at the effects of 518 and pharmacokinetics, and then CSF sampling. So just as I mentioned before, it's important to get the drug into the blood. It's equally as important to get the drug into the CNS. And CSF sampling will allow us to see what happens in the plasma compartment around the brain. So it's really important for us because this confirms target engagement. So I'll show you the results. So in the single ascending dose, which is on the left-hand side of the screen, what you see is you can see the placebo all the way up to 135 milligrams. As we increase the dose, the percentage of baseline HTT RNA goes down. This is important because we looked at, again, over time being titratable. So this shows the titratability, that as we increase, there's a linear dose response. And then we also looked at this with regards to the SAD data. This was a one-time dose, 24 hours. And so we took we looked at the values in the blood at 24-hour post-dose. And again, what we saw is we saw this nice curve. We saw this nice decline in the percentage of HTT RNA, which for us indicates that we would expect to see at some point in time the lowering of the HTT protein. This allowed us to pick doses and move into the MAD cohort, so the multiple ascending doses. So this is going to be multiple doses over a time period of 14 days. And so what we studied is 15 and 30 milligrams. And so this allows us to see multiple doses over time, what happens with the trend. And you see the same as you increase the dose and as you increase time, you see this reduction of the Huntington RNA. This is incredibly important when I show you a couple slides here in a second. But this confirms to us that we can do it in multiple doses, that there is a nice response that we see with these patients or with these healthy volunteers in the drug. So here's the other component. So we have the titratability on the previous slide. Now we want to understand durability and reversibility. So there are 2 concepts, and I think they're important here, is when you look at the SAD dose, and this is the 90 milligram dose on the left-hand side of the screen, what you see is as the drug comes up in concentration, you see the percent of baseline HTT RNA go down, and you see this nice trough then at 24 hours. So again, this is what we're looking at, 1 dose, 24 hours, and then we want to understand what happens with this tail of this curve. So you can see in the gold orange line that after 24 hours, it does go back to baseline. But what's nice about this is the durability of effect is up to 72 hours. So even though I don't have the drug in my system, I'm still seeing this continued suppression of the mRNA, which is important, I think, in the long-term treatability of Huntington disease. So this for us is important. This is incredibly informative for us. So what we do is we look at this in the MAD. So the same concept. So this is after 14 days of dosing. What we look at is what happens after we stop dosing, take the foot off the gas, if you will. What happens with the momentum that we've built up? And we see that it is reversible, so it will go back to baseline over time. And we do see this consistent splicing still occurring. So the lowering of the mRNA is durable and consistent in the MAD doses. So this is important. And as you can see here, the 15 and the 30 milligram, you saw more lowering in the 15 or in the 30 as compared to the 30 milligram or 30 milligram as compared to 15. So this is important because we then do see both of the components that is titratable and is reversible and durable. So this is important for us. As mentioned before, so the other component of the study is we're looking at the CSF. So here we have the human plasma CSF correlation, and we see then the non-human primate CSF correlation. And what we see is in humans, we see that we do get target engagement, that the drug is crossing the blood-brain barrier, and more importantly, it's staying in there. So it's not being actively effluxed, that it is staying in that area. So this is important because as we're treating HD, we're treating this as a whole system disease. So this means we want to make sure we get there, we stay there for the maximal opportunity of success for the patients with HD. As I mentioned before too, so this is the protein data. So when we look at this, and this was a cohort we looked at in our MAD study, so multiple ascending dose, this is cohort 2-3, and this is after 21 days of dosing. So what's really important is we see the Huntington mRNA comes down and it reaches steady state in about a week. So that means that we have the maximal lowering of the Huntington mRNA. What we see and what we know to be true is the half-life of the Huntington protein is longer than the half-life of mRNA. So as expected, we do see the lowering of the Huntington protein. As I mentioned before, these go hand in glove. But what we do see is it does take a bit longer to lower the Huntington protein as compared to the Huntington mRNA. So what the natural conclusion is and what we really feel strongly about is, yes, the drug works. Yes, we get target engagement. And yes, we get the results that we're looking for here. We do need to go longer, though. So in order to get the maximal steady state for protein reduction, we do need to go longer. But this, again, this is success for us. This is important for us because this is really what we're designing the program to do and to look at. So when we do the simulations, so one of the ways that we can look at this is we can simulate what should happen, and this helps us to understand what we know about the half-life of mRNA and half-life of protein. And what we see is we do see we're reaching, I'll overlay the real data on here, so this is a little bit easier. What you are seeing is that the simulation shows what should happen with the mRNA, and then we see the PTC 518, which is the purple dots. We do see it behaving the same way that we would expect in the simulation with regard to mRNA. And so this allows us to extrapolate out and understand what the curve should look like for protein abundance. And what we do see is that it's tracking on target with what we'd expect. So this allows us to be confident that we say we should see, protein lowering within about 6 weeks of the lowering, after administration of the drug. So this is important because, again, it helps us to validate the next targets as we move into the next phases of development. So what we're doing in the phase 2 study, there's really 2 things that we want to target here. Again, this is exciting because now we're into HD patients and now it gets real, if you will. So what we're going to do is we want to see the tolerability and pharmacology of 518 in Huntington mRNA and protein reduction. So those are the two key components. We also want to see the effect in blood-based CSF-based and radiographic biomarkers of Huntington disease. So this is going to be what's the pull through. What additional benefit do we see in these patients? So the way the study is set up and designed, it's a double-blind, multiple-dose, 12-week placebo-controlled study with a long-term open-label extension. So what we have here is at baseline, patients will be randomized to one of 3 treatment arms, either placebo, dose level 1, dose level 2. We're still adjudicating which doses those will be. After they complete the 12 weeks, then they're going to be randomized into the open label extension for a year. So what this will look like is, again, we'll drop the placebo at that point in time, and then they would be on dose level 1 or 2. So this is important because this gives us long-term exposure. We'll see what happens in the 12 weeks to evaluate the Huntington lowering as well as then the ultimate protein expression. So this is important. This is an exciting study for us because one of the things that we're looking at doing is looking at, as mentioned, the endpoints are here, safety and tolerability and reduction of blood, but we want to look at biomarkers. We want to understand things like the Huntington protein and CSF, the NFL changes, and also we're going to be looking at volumetric MRI. So we'll look at caudate putamen and ventricular volume changes over time with treatment. And we want to see the correlation with clinical endpoints. So what changes? Typically in ADHD, what moves early are things like motor and cognition. So we'll be looking at those in particularly as we study this in HD patients. So the important thing that we've seen is as we've looked at natural history and as we've looked at tens of thousands of patients and really followed this, we're using natural history to understand what the progression looks like, and more importantly, to identify the things, as I mentioned in the previous slide, what moves first and what is modifiable. And also this helps us to determine the target population and really select endpoints which are meaningful to the patients, meaningful to the clinical benefit, and really helping us to understand what we're doing with regard to trajectory and change. So it's important for us, and we feel very strongly about this, that we have to pick the right patient populations, intervene earlier. As we've talked before, and as I've mentioned in other presentations, when neurons die, neurons are gone. So it's hard to resurrect those. So what we see is typically in the old stages of clinical development and old studies that have been done, a lot of times by the time you see TFC changes, and this is total functional capacity, by the time you have functional capacity changes, it may be too late sometimes to intervene with certain disease-modifying molecules like a 518. What we want to do is in our studies, we want to look at the target population that has active disease. So that means we have volumetric brain changes. And we're beginning to see changes in things like SDMT, which measure cognition, and TMS, which measure motor function. These tend to go first before you have major changes in functional capacity. We really feel this is important because we want to preserve as much of the function capacity, and we want to preserve as much of the brain tissue as possible in the HD patients, because we know this doesn't get better with time, and we know that naturally untreated this will continue to progress. So we want to intervene. We want to change the course of disease progression. We want to change the outcomes for these patients. We feel very strongly about this. We believe that this is the population really to target. So this looks like, just in conclusion, when you look at the inclusion criteria that we have 25 and older, ambulatory. We're looking at CAG repeats of 42 to 50 inclusive, and then we're looking at different parameters such as volumetric changes, total motor score, cognitive score, and we're using something called a prognostic index of Huntington disease. So this is a factor by which we can understand patients, as compared to natural history, have a higher likelihood of declining when they fall into certain risk categories. And the PIN is just a way to look at this risk category and look at that disease burden. So this is really important because I think this allows us a peek into the window to understand who should change and when they'll change. And again, get just-in-time treatments for these patients to change the velocity of decline. We're excited about this. It's incredibly, earth-shattering in a lot of ways for us, and innovative, if you will. We really want to change. We want to pivot away from what people have done in the past, and our study is the Pivot HD trial. So we mentioned that because we want to learn what other people have done, what other companies have done, and we want to do it differently, and we want to do it better. So in closing, the main components that we see necessary within the HD drug development piece is they have to be orally bioavailable. They need to cross a blood-brain barrier and distribute evenly throughout the brain. Need to have enough free fraction in the blood and tissue for target engagement. So it has to be in the CSF, and it has to stay in the CSF. It has to not be effluxed. And then we want to make sure that we see the correlation between mRNA and protein lowering, as we've seen in healthy volunteers. We want to see this then in HD patients. We want to see, as I mentioned before, Huntington lowering across the different areas of the brain and peripheral tissues. We would like it to be reversible and durable, and we want to make sure that all of these things line up to help us really put together the best clinical development program possible. And we feel incredibly confident at this point that we have done that. And again, it's been my pleasure to talk to you about this and give you updates. I definitely hope to talk to you more in the future. And again, good luck, and thank you for all of the HD community, for everybody coming together and really pulling for better treatments and better options. This helps the development process. It helps everybody. I'm just proud to be part of the HD community. Thank you so much for your time, and have a great day.
Hello, can you hear me?
Yes.
So I'm Chris Ross, and I and Blair Levitt will be moderating the discussion and question period. I think we had a very exciting series of talks with really remarkably diverse and interesting mechanisms, different ways of approaching HD. Even though we have the failed Roche trial recently, this shows what a panoply of new and exciting approaches we've got. And I should mention, by the way, that this traditionally has been a session in which we have the companies that are a little further from sort of definitive clinical trials, but many of the phase 3 studies that, for instance, that you're hearing about today and that have happened in the past have been in the Innovators Forum first. So hopefully this is kind of a stepping stone to larger things. What we thought we would do to get the questions going, I see a number of questions coming in already, is kind of do one question as what's called in U.S. The political Sunday morning talk shows, a lightning round. That is a single question asked to all the speakers, ask for just a 2 or 3 sentence answer to kind of get us going and get us thinking. And Blair is going to ask that question. Blair, I can't hear you. There we go. Can we take the slides off? Yeah. Can we take the slides off and just put the people up?
All right.
Go ahead.
You can hear me?
Yes. Good.
So you have a captive audience here of the professionals who are involved in studying Huntington's disease for many years and doing HD clinical trials. This is your opportunity to tell us, what is the one thing that the HD community can do to help advance your specific program? Another way of thinking about it is, what's the major roadblock that we can help with? So I'll give each of you a time to answer the question, and we'd love to hear what we can do to help move your programs forward.
And let's go in the order of the way people spoke.
Hi, so I'm Dietrich. I'd answer that by saying, in the post-Roche phase 3 world, I think a lot of big pharma companies have slowed down their interest in Huntington's disease. At least we're seeing some of that in our partnering discussions. And I think if HSG would be willing to facilitate and broker those types of relationships, it could be catalytic, at least for some of the smaller preclinical companies like ours.
Okay, but what would you like to know about HD is what Blair is asking. What information?
So emerging non-invasive biomarkers, I think, would be exciting for us to get a handle on early.
Great, thanks.
I believe I'm next. I'm Russ Addis from NeuExcell. I think it's a great question to understand. From my perspective, it would be to understand more about the timing of progression in patients as relates to potential efforts to reduce the mutant Huntington protein. Is the best that we can hope for at certain stages just a halting progression, or are there stages at which you might be able to really restore function? I think that's something that is really challenging to figure out in animal models. So that would just be wonderful to get everyone's perspective on how we can advance that knowledge.
Great, thank you. Ophidian?
Yes, this is Doug Mcneil at Ophidian. I think one of the things we're interested in is as detailed a database of the community as possible. What's their natural history, as much as you can put together, with any studies that have been done on their expansion, on the number of CAG repeats, on biomarkers, so that when we design a trial, we start designing from the most information and not have to go out and sort of accumulate that in the patient population, have a database that we could design our trial based on what we think will be the efficacy markers of the trial.
Great. Triplet?
Yeah, hi, Irina Antonievic, Triplet Therapeutics. So thank you for this question. I find this really exciting. So given the triplet target somatic expansion, my dream would be to have a way to measure brain somatic expansion somehow in the CSF or in some other way that gives us an insight into brain somatic expansion. This would be, of course, fabulous and might be very difficult, but it would be fabulous. And just one quick other addition is a functional benefit in a mouse model of somatic expansion and halting it. This is also something that I think the field has not yet been able to demonstrate, and it would just corroborate, I think, the human genetics findings.
Great, thank you. PTC.
PTC.
Yeah, so I think this is a great question. I think one of the things that we're looking at that would really be important is within the HD community, having the patients and having the patients to let the studies run, right? So both having the participation, understanding what moves early, having definitive markers which indicate treatment success, and ultimately being able to see this through to fruition. So these type of meetings are helpful because we see what everyone else is doing. We can share our thoughts and ideas. So that collaboration, I think, is important.
Great. Thanks very much.
Great.
You know, what I'm really struck by is a number of the questions really involve the natural history of HD. And I would argue that HD is one of the neurodegenerative diseases that we have the best natural history information available. And yet, there's still so many things we don't know, like when do cells begin to die versus have dysfunction. And actually, I will ask this as a question to Irina from Triplet. When do you think the somatic expansion begins to happen? For instance, might it be a developmental process, in which case it might be too late?
Yeah. And this is, of course, the question that we have asked ourselves, and I can only answer it based on the data that exists in the literature. We do know that it is not present at birth. So this, we do know that it starts sometime in life. And some of the postmortem brain data from HD individuals who died prior to motor onset or prior to any obvious symptoms, they do show that somatic expansion starts in the striatum about 13 to 15 years prior to motor onset. And so because we know that at the time very few cells are affected with somatic expansion, we think this is the right time to start to intervene to prevent further expansion and thereby prevent the degeneration and sort of rescue or maintain the function that is present at that early time.
Great, thank you. Let's see, so we had a question.
Chris,
let me ask one more, Blair, and then you go. We had a question from Southwell about the target of the Neubase. So you showed kind of a, I think you called it a conceptual example, but I guess we would like to know more about exactly what is the compound and what is it targeting.
Sure. So the compound is a peptide nucleic acid that targets the expansion directly and inhibits either translation by a ribosome, if we're targeting the mRNA of the mutant allele more selectively, or inhibition of transcription if we're targeting the genome. We have various modifications of the compound that we're working on that can actually see not only the single-stranded structure, for example, the mRNA of the expansion, but target the double-stranded structure, which is relatively unique, at least relative to the wild-type transcript. So hopefully that answers your question.
Well, let me actually ask a follow-up then. You said yours is ultra-high affinity, quote-unquote, and I guess I'm just wondering, sort of briefly, chemically, why is your peptide nucleic acid better than the kind of previous antisense oligos or other related compounds?
There are two reasons. First is it's neutral in charge, and prior generations of ASOs are generally negatively charged. And so as they approach nucleic acid target, the backbones repulse one another and results in a lower binding affinity as they hydrogen bond. So number one is neutrality, so the backbones don't oppose one another. The second is the molecules were actually shaped, so they have a helical shape to them, which reduces the free energy necessary to convolute themselves to engage the target, and that contributes to the higher binding affinity. And you can actually dial those in and get selectivity for either mRNA or DNA through backbone modifications.
Great, thank you. Blair, why don't you go ahead?
Yeah, this is really a question for all of the Huntington lowering groups. So Irina, you can relax for a second. You don't have Blair's question to answer. There was a suggestion that systemic delivery is going to be better, and there's no question that as a physician, it's easier to deliver systemic than central. And there's even a suggestion that perhaps it would be beneficial to lower Huntington in the periphery, but animal model and animal studies have suggested that peripheral lowering of Huntington can actually be harmful and induce side effects and toxicity in different organs. Even if it's relatively mutant Huntington selective, you're going to have higher levels systemically than you have in the brain. And so that relative selectivity is going to lead to wild-type Huntington lowering. So all of those of you who are proposing that systemic delivery is better, how are you going to deal with the potential toxicity of targeting wild-type Huntington in other organs? And maybe I'll direct it first to Dr. Stephan, since he was the first to raise this as a benefit.
No, thank you, and I hope I can accurately represent my colleague Robert Friedlander's thesis on this. We do believe mutant Huntington protein in the periphery is pathogenic in various tissues, and the simple answer is dial in the selectivity as best we can, quite frankly, and enrich as best we can for CNS penetration versus uptake and exposure in other peripheral organ systems. And so that's the balancing act that we do on this program internally.
Dr. Mcneil?
Yeah, I think as I mentioned, we actually, although given systemically, we target preferential delivery to the brain by targeting receptors that are only present on the brain and counter targeting receptors present on peripheral. We enrich for delivery to the brain. So we've kind of thought about that as well, and it's almost yin and yang. We worry we won't have enough knockdown of mutant protein in the periphery, but we're looking at probably 5 to 8-fold enrichment in the brain, so there'll be some in the periphery, but the predominant action will be in the brain.
And maybe someone from PTC might want to weigh in on this as well.
Yeah, so I think that the approach that we have is we're partially lowering it. And so one of the things that we want to understand is, by taking efflux out, will we have a similar lowering between CNS and periphery? So without the complete lowering and complete obliteration, of the Huntington, I think that we should avoid some of the peripheral side effects. We'll monitor the patients through the trial. And really, keeping in mind in the uniform lowering component of this that we don't think we'll run into an issue, but we'll keep an eye on it.
All right, Chris, you want to go next?
Yeah, sure. So let me pass on a question from Roby Blumenstein to NeuExcell, and the question is: are you considering a combination of glial conversion plus some kind of Huntington lowering type therapy? And I can probably think of some follow-ups to that, but why don't we start with that?
Sure. The simple answer is yes, we are, and that's something that our partner Spark is certainly looking into as well. I will comment that and this was related to one of the other questions that came through. We are targeting astrocytes in the HD brain that would almost certainly contain the same mutation that neurons would have, and so yes, we certainly are aware of the fact that, or cognizant of the fact that that our approach is not going to directly reduce the mutant Huntington. I will, however, point out that we have looked histologically at the newly formed neurons that we generate, and we see greatly reduced levels of aggregated mutant Huntington protein in the newly formed neurons. Exactly why that is, we'd love to try to figure out, but I think that is something that's worth noting.
Yeah. Well, let me follow up about that, because I think everyone was really impressed by the phenotypic data that you showed, and I think that raises a mechanistic question, because we normally think about these HD model mice as not really being so striatal specific, and so I guess there's two parts to my question. To what extent do you think the effect is dependent on the striatum? And also, to what extent do you think that the neuronal conversion within the striatum results in functional circuitry, for instance, cortex striatum globus pallidus?
Right. Yeah, it's a great point. I mean, the new cells that are generated are restricted to the striatum. That is where we're directly administering our AAVs. But we have looked at connections, and we do see, tracks tracing all the way down to the globus pallidus and other appropriate structures. So there is definitely some interconnectivity that's happening there.
Okay, but do you see synapses?
Yes, we do. Yeah, we could.
Very good.
Yes, we do. Yeah.
Okay, Blair.
All right. There's a couple of questions here for PTC. Specifically, is the IND accepted by the FDA for the Pivot HD study? And apparently it's not up on clinicaltrials.gov. Is it going to be a U.S. study or a global study? And just as a complete question, when do you envision starting your phase 3?
So I'll actually let my colleague Brian Beers discuss this. He's the expert strategist, so I'm happy to have him address that.
Sure, no problem. I can say the IND has not been accepted yet by the FDA, so we're still working on that. The phase 3 study, I'll take that question now. So, a great question. I wish I had a better answer to that than I do, but I really don't know. It's highly dependent on phase 2 and what we see coming out of phase 2. I can have to underscore the importance of doing a proper phase 2 study and getting dose right before we go into something like phase 3. So it's of utmost importance. When we do have the phase 2 start date, the trial will be up on clinicaltrials.gov.
Great, thank you. Let me ask you
Great,
another.
thanks, Brian. And Chris, I'm gonna.
Yeah, go ahead.
Sorry.
Blair, you go.
Go ahead.
Well, I was going to ask.
I said I was going to pull a Chris Ross and do a follow-up question.
Oh, go ahead, please.
The other Brian. This is another Brian question. So quickly, you're altering splicing, and we know where you're altering splicing or where you're targeting splicing, but we all know that there's at least the potential of a pathogenic exon 1 splice fragment, pathogenicity not yet proven, but is there a possibility that altering splicing further down may increase the levels of a potentially pathogenic exon 1? And how do you think about that, and what have you done to look for that?
Yeah, so one of the things I think that we look at is we think about the levels that we'll be dosing at. And so when you look at it, we're really targeting HTT at the levels that we'd use for the efficacious doses in the trials. The downstream that you'd be talking about would be at much, much higher doses. So I don't think we're going to see that, like tenfold from where we would be in the dosing. So, we're not overly concerned about that at the moment, but we'll definitely keep an eye on it. And we don't anticipate it happening. However, with due diligence, we'll keep an eye on it and see if we saw anything.
Okay, I'm going to ask a follow-up question to Blair's follow-up question, and that is, so you're affecting splicing. Are you affecting the same exon that the Novartis drug does, and what are the differences and potentially the advantages of your drug versus the Novartis drug?
Yeah, I think one of the biggest advantages that we see, and I mentioned before, is we're specifically targeting HTT in Huntington disease. So the branaplam product from Novartis was used in SMA originally. So when you look at the specificity of the splicing, I think that the HTT is really much more specifically spliced with our molecule. We're not anticipating some of the challenge you have with a more broadly spliced molecule.
Okay, I may have missed part of that part of your talk, and it is targeting the same exon, is that right?
Yeah, and I think the other component that you see within branaplam that's different than ours, branaplam is actively effluxed. So for our molecule, we don't have the same efflux. So once we get into the target tissue, we're going to stay there and have that benefit. Looking at that uniform lowering, is really going to be, I think, a key component, differentiating factor with our product.
Great, thanks. Blair, you want to go again?
Great. Yeah, sure. I have a quick question for Irina. I don't want her to feel like she's getting off the hook here. Irina, so you've presented really the first approach to using ICV administration in HD. We have a question about whether the drug could ever be administered systemically. So that's one question. But also with the first use of these ports in an HD population, will that require specific safety studies of the mode of administration in addition to administration of the therapeutic?
So let me start with the second question first. We don't envision any particular safety studies first. We had regulatory interactions where we have proposed to implant this catheter, which is a standard procedure. So it has been done for more than 60 years in many patients. It has been used to administer drugs for over 19 years in certain patients. So there's a lot of experience with this kind of devices in humans in different indications. And so we did not have any particular questions about these devices. So we would test the device in the combination with our drug in a phase 1 trial where we would have a placebo control. So we would evaluate our drug against a small placebo cohort in this multiple ascending dose trial. So this is our current plan. In terms of the systemic administration, right now our molecule is an unconjugated antisense oligonucleotide, and these molecules do not allow to be, we can inject systemically, but it will not reach the brain in sufficient concentrations. So we are looking at follow-up modes to, once we have shown that lowering MSH3 by about 50% is safe, well tolerated, and has a therapeutic benefit based on biomarkers and clinical outcomes in the initial trial, we would start to look at other ways of doing this differently. But for now, I think an antisense oligonucleotide will have to be administered into the CSF unless it's conjugated to something.
But I guess that a follow-up question to that, and this is, I think, in part Kenneth Servin's question, is you have a small molecule program. Is there any possibility of small molecule targeting of this target?
So, and I'm not a protein chemist, so take this with sort of like some grains of salt. But what I have understood is that targeting MSH3 with a small molecule is really, really difficult. There have been multiple efforts. I believe the CHDI has actually done some effort, has looked into that. So it is a difficult protein to target and to really abrogate the function. But one of the aspects of a small molecule for MSH3 is also that this is a molecule that we do want to have in the brain, because somatic expansion, even though it does occur in the liver, it's not ubiquitous. It is in certain areas of the brain also only, and maybe in the liver. And so we would actually think that a safety profile is better if it is targeted to the brain areas where it really has the effect of halting somatic expansion, but does not really have a whole body effect.
Great, thank you. Blair, you want to go? I've got something if you don't.
You can ask the next one, Chris. Go ahead.
Well, let me do another lightning round, if I could, again in kind of the order in which you spoke, and that is a number of you talked about using biomarkers, and I guess what I'm curious about is how are you thinking about biomarkers in terms of treatment effect and in terms of the fact that the FDA recent activities in Alzheimer's disease notwithstanding, the FDA normally does not accept biomarkers as an outcome. They want to see a functional or something, feeling, surviving, etc. And so what are you all thinking about that? And just kind of a brief answer, if you could, and we'll start with Neubase.
Yeah, I think we still have time before we get into the clinic, and we're hoping that, for example, some non-invasive imaging with specific HD-based ligands emerges as, a valid biomarker strategy and is accepted by FDA. We're hoping to draft off of others on this topic, and so we don't have a specific program to invent new biomarkers or present the case to the agency.
Yeah. Okay. Good. NeuExcell.
Yeah, we're certainly very interested in biomarkers, imaging, of course, but also looking at both doing some proteomics analysis in both CSF and perhaps blood biomarkers, really more exploratory at this point, but anything that we could find to help us understand the process would be fantastic.
Yeah, I think the concern with anything that's injected into the striatum is, how would you use imaging as an outcome, because you'd be altering the thing that you're measuring. But let me leave it at that. Ophidian, any thoughts about that?
Yeah, so mostly the biomarkers we're thinking to help select the dose with, both early on, not as an efficacy marker per se, and would have to do the long-term studies to show behavioral and cognitive changes that are necessary to halt, slow, or reverse the disease. Imaging, though, may fall in between as an early measure of efficacy.
Yeah, good. Triplet, you've thought a lot about this, I think.
Yes, we thought a lot about this. So obviously my wish is to have a somatic expansion biomarker, which would be great. But we have developed our own target engagement biomarker because we are not targeting HTT. And so this will be, of course, very important in phase 2. I also think that imaging biomarkers as well as neurofilament light will be important biomarkers in phase 2, at least to give us, the confidence that we are really changing the disease trajectory. But I absolutely hope that as part of the CPAS consortium, as a group of HD clinicians and developers, that we can make some inroads with the regulatory agencies to at least sort of have a dialogue about biomarkers, maybe, like with aducanumab, accelerated approval, but then continue for a clinical outcome. I do think in a disease with such a high unmet need like HD, this would be, of course, very, very important. And I think I hope that as part of this dialogue, we can maybe move the openness to accept a biomarker.
Yeah, no, I have to say that's something I'm very sympathetic to, that as we know more, maybe the outcome measures that the FDA will accept will change, and I think that was Neubase's thought also. PTC, you thought about this a lot also.
Yeah, so I'm kind of a belt and suspenders guy, right? So if I want to keep my pants up, so in the clinical trials, I want to design a trial that's got clinical endpoints, but I also want to see the biomarkers. I want to see the clinical correlate, and I also want to move away conceptually from the idea as brain tissue as a biomarker. An organ is an organ is an organ. So I really like to see some movement there, because I think this is critically important that HD doesn't get better with age, nor does brain tissue come back when it's dead. So I'm very adamant about this for the patients, that we need to make sure that we look at everything possible, position ourselves in a position to really be able to bring this to patients to really change trajectory. Belt and suspenders.
Yeah, thank you. So I think the message which many of us would agree with, and certainly the FDA would love to see, is that you want to have a whole story. You want to have a clinical outcome that's good. You want to have good safety. You want to have biomarkers that are consistent. You want to kind of have a whole package. Great. Okay, Blair, you got anything else? I think most of the questions. Yeah, go ahead.
Oh, I was just going to follow up on Irina's point, and the HDRSC is an excellent group that is focused on developing better clinical trial paradigms and biomarkers and modeling for Huntington's disease, specifically to advance therapeutics. So those of you who are maybe earlier stage companies or not yet participating, it's never too early to work together in this sort of non-competitive space to help develop this. And I completely agree with the sentiments that improving our measures for clinical trials is something the HSG is committed to, and the HDRSC is also advancing both our understanding, but also interactions with regulatory agencies to help ensure that when we develop these biomarkers, they do help move products into the clinic and ultimately into approval. So second that emotion, Irina. We should all be working together to do this.
Yes.
Yeah, especially
maybe on that happy collaborative note, we can.
Yeah, and especially.
Thank you, everyone, for your excellent talks. This was really exciting.
Great. I think we've pretty much covered it. I think we've answered, or at least addressed, pretty much all the questions from the audience, as well as having our own little discussion here. I think this has been a great session, and it really shows the variety of different approaches for this devastating disease, but that has lots of different possibilities for it. So I think we can wrap up at this point.
Thank you.
And thank you, everyone.
Thank you very much.
Thank you.
Bye-bye.
Bye-bye.